Okay, so today I'm going to be going over the Lytic and Lysogenic cycle. I did kind of go over this in a separate video, but I'm going more in depth on this. So it's important to know that these cycles are specific to bacteria. These will only happen with bacteria. And the Lytic cycle eventually leads to cell death. So this is when the cell is going to burst out with new phages, infecting other bacteria, repeating the cycle. So the first step would be attachment. Attachment is exactly what it sounds like. It's when a bacteriophage attaches to specific receptors on a bacterial cell, and then it'll inject through penetration, it'll inject its viral genome. Third step would be biosynthesis. So biosynthesis is when the viral genome is going to take over the host cell's machinery and command it to make and replicate a bunch of its viral genome, a bunch of its viral DNA. It'll also command the host cell's machinery to create proteins that'll kind of be the phages, capsids, coats, tails, etc. And step 4 is maturation. Maturation is exactly, again, what it sounds like. This is when the cells or the phages, are maturing. It's like an assembly. So everything is kind of coming together. Phages are getting their capsids, etc. Step 5 would be lysis. So this is when the bacterial cell is actually going to burst out with all those new phages. This will lead to a bunch of new phages scattering out through the environment, looking for more bacteria to infect, repeating the cycle. This is the active state of kind of phage replication within bacteria. The next kind of phase we have is the lysogenic phase. So the lysogenic phase is more of a dormant phase. It's more silent. So the first step of the lysogenic cycle would be attachment, which would then lead to penetration. So again, attachment would happen through the bacteriophage binding to specific receptors onto the bacterial cell, and then it will inject its viral genome into the bacterial cell itself, which leads to integration. So integration is essentially when the viral genome combines or integrates itself with the bacterial cell's chromosome. Now that the genome is integrated with the bacterial cell's chromosome, it'll go through many phases of binary fission with that viral genome now incorporated into its chromosome. So it'll go through many phases of binary fission carrying on that viral genome. A bunch of new bacterial cells will obviously contain this viral genome, and it may go on like this until the cell dies or whatever happens, but essentially what could happen is induction, and induction would lead to the Lytic cycle. Now induction, like I said, may never happen, but if it does happen, it's usually because it's a result of stress, environmental stress, whether it's from heat, chemicals, pH imbalance, UV light, radiation. Essentially what would then happen is the prophage gets cut out of the chromosome, thus beginning the Lytic cycle. Don't think that the Lytic cycle is always a result of the lysogenic cycle. It can only happen through induction. But the next thing I kind of want to talk about is how animal viruses multiply. Same kind of idea through attachment, entry, etc. The biggest difference is the entry. So with attachment, the virus will attach to, again, specific receptors on the cell. Entry can happen in one of two ways. It can happen through fusion, which is through primarily enveloped viruses, and then there is endocytosis, which is for non-enveloped viruses. So fusion happens when the membrane or the envelope on a virus binds or kind of integrates with the host cell's membrane. It kind of fuses with the host cell membrane. Thus, the viral genome is getting incorporated or getting inserted into the host cell. Endocytosis is a little bit more complicated, I guess. That is when the host cell essentially engulfs the virus itself. It's a little bit more trickier than that though. First, need the formation of a membrane vesicle. So when the virus binds, the cell mistakenly thinks it's something that it should bring in to the cell itself. The cell membrane will kind of invaginate, it'll fold inwards, bringing in the virus through the membrane vesicle. After either one of these processes, though, the virus will then get released into the cell cytoplasm, which leads me to encoding. Encoding happens when the capsid gets broken down, releasing the viral DNA or RNA into the host cell. And then biosynthesis, the virus will, of course, hijack the host cell's machinery, replicating the DNA, proteins, etc. Maturation will happen. That is when everything is kind of getting assembled together. And then release is when everything is going to burst out. Now, release can happen in one of two ways. Again, enveloped viruses will be through budding. So the virus or the phages or whatever will get pushed out through the cell membrane within the host cell through little pores or little openings within the membrane. As it gets pushed out, the virus will take the host cell's membrane and kind of, break off a piece of it and take it with them, that actually becomes their envelope. And then for non-enveloped viruses, it'll just burst. It'll cause lysis. It'll just burst out of the host cell. And then I kind of, like I showed you earlier, I kind of illustrated it here. So fusion, you can see the cell membrane is fusing with the virus's envelope, releasing the DNA. And then endocytosis is actually when the virus gets engulfed into the cell membrane. I hope I made this very simple to understand.